So good afternoon, everyone, and nice to see some familiar faces. And I'm really here to kind of extend on my talk that I gave last year. So for those of you that were here before, this will kind of just, I'm going to give you a little bit of a refresher. And for those of you that this is the first time, I'm going to give you a nice little recap of where we are. So proteomics. Very fancy term. What does that really mean? So let's recap. So this is a very simple diagram that kind of describes how the instructions within your DNA is converted into a protein, right? So we call the study of DNA genomics, and we call the study of proteins proteomics. So it's as simple as that, really. So what are proteins, right? So proteins have a variety of functions. So, and I'm kind of just summarizing a few of them here, but they're important in catalyzing reactions inside the body. They're important in fighting infection or as part of your immune system. They're structural proteins that act as kind of scaffolding inside the cell. They're important for transporting molecules inside and outside of the cell, and they're important for as kind of messenger proteins, kind of what we call cell signaling, kind of telling a cell what function it needs to execute or if it needs to talk to other cells in the body. So those are, broadly speaking, functions of proteins. Now, why we are interested in proteins is because proteins can act as biomarkers. Now, this is just, again, a technical term for really, a biomarker is a marker of disease. So where we can look at your cerebral spinal fluid, at your blood, feces, urine, and we can identify this marker, this biomarker, and that can tell us something about the disease. So there are a variety of different biomarkers, and the ones that we are particularly interested in are diagnostic biomarkers. So in other words, do you have the disease, yes or no? Or prognostic biomarkers, so biomarkers that can tell you something about what is to come, right? So the disease progression, are you going to progress quite quickly with your Parkinson's, or are you more likely to develop dementia? So these are the kind of questions that we want to ask of our proteomics data. So this is just a reminder of the project itself. So we are taking your blood that you have very kindly donated to us, and for a small set of cerebral spinal fluid, and we're putting it through this very advanced high-tech platform that is made by this company called Olink, where we were able to test over 5,000 different proteins and give it kind of a relative quantification of those proteins. And then we analyze this data, and we're talking about over 20 million data points. And with that, we are hoping to distill some kind of signature of Parkinson's disease. So, where are we now with this project? So, last time I showed the slide, we were in analysis phase 1, and now we are in analysis phase 2. And here are some of the tantalizing results that I can share with you today. Just a note before I continue is I'm going to focus mostly on blood. So all the results you see, I'm kind of referring to results in your blood. Right, so what you see here, these are called volcano plots. Technicalities here aren't too important, but essentially what this is visualizing is what proteins are increased and decreased in somebody with Parkinson's disease or REM-sleep behavior disorder compared to healthy controls, right? So if it's in the blue box, that means those proteins are decreased in the Parkinson's group or the RBD group compared to healthy controls, and red is increased, right? So the first thing to note is that there's an overlap in these proteins, right? And this is a reassuring finding and something that on some level we would expect or hope for, because we know that PD and RBD are linked, right? So we know that there's a biological link between these two conditions through what we've been demonstrated before and discussed earlier today about seeding amplification assays that we can identify alpha-synuclein in PD, and we can also find that in RBD. So already we know there must be on some level a biological link, and we're seeing that in our proteomic data set. And when we try and kind of dig into what do these proteins actually do, what are their function? And I want to preface this with saying that, unfortunately, we still don't really know the function of a lot of proteins. We have vague ideas of a lot of them, and we can kind of broadly classify their functions. But when we have to kind of in detail describe the function of every protein that we have in the body, and we're looking at a minimum of 30,000, maybe 5% of them we have a fair understanding of what they actually do. But that being said, we have broad understandings and kind of we can class them into groups of what they've kind of been linked to in the research. And these proteins that are kind of shared between Parkinson's and RBD are what we call are enriched for or are relevant in immune activation and cell-to-cell transport. Now, immune activation is a recurring theme, as I'm sure you've noticed today. And again, this is something that we find promising to see that that's something that's coming up in our data set. And cell-to-cell transport is also within kind of the paradigm of Parkinson's a finding that kind of fits, but how exactly it fits is something we're still trying to figure out. Now, the trouble with Parkinson's, however, and doing a project like this, is that Parkinson's is not the same for everyone. I mean, you would know even better than I do that Parkinson's is different in every individual. And when, in the ideal world, when we do a study, when we do, comparing apples with pears, when we say compare Parkinson's with healthy control, we would ideally want a very clear, distinct dichotomy between the two groups, right? So in the ideal world, Parkinson's is this one homogeneous disease. But unfortunately, in reality, it's like very different for everybody, right? And the term for this is heterogeneity, but essentially just means there's high variability in the expression of Parkinson's disease. And the trouble with that is that when we do this direct comparison, PD versus healthy control, we're really just looking at an average signal, right? We're taking all the PD people and we're just saying, what is kind of, on average, different in this PD group versus the healthy control group? But that kind of loses out on the fact that there's this huge variability within Parkinson's. And this is something that, we've done quite a lot of research on, this group has done a lot of research on, and this is maybe a diagram that you've seen before, but this is kind of work that's been done previously that tries to capture that heterogeneity, that variance within PD, right? And the way that we've described that is we've clustered into these four clusters that are purely kind of driven by clinical features, so your degree of motor symptoms, your non-motor symptoms, your response to treatment, your progression. And as a kind of rule of thumb, broadly speaking, is cluster 3 tends to do the worst. So I'm going to kind of quote that as like the more severe version of PD. And cluster 2 tends to do the best, right? And that kind of captures already this kind of variability in PD. So now the question is that also expressed in the proteome? Do we see those differences in the proteomics? And we do. So essentially, the first thing to note is that we as a kind of a sanity check, we can look for proteins that we expect to be different between the groups because people are on different doses of medication. So someone with a milder version of disease, cluster 2, will be on less medication. And as a result of that, the protein called DDC, which is at the top there, I don't know if I can actually, if this is going to work, yeah, here, and prolactin, which is down there. These proteins kind of change in response to treatment, and they change in the expected direction that we would expect them to change, right? So that's already kind of a sanity check. So, okay, we can kind of see a protein difference between these two groups based on their treatment. But there are other interesting proteins that come up as well. So the one is something called this GHRL, which is ghrelin, which is a hormone that drives your hunger, essentially. And this is a protein that's kind of been flagged quite recently, and work is being done by PPI, the big Parkinson study, global Parkinson study, that's looking at if ghrelin is neuroprotective. Right? In other words, if you've got ghrelin or lots of it, or it's normal, let's say, then your disease progression will be slower, your PD will be better, versus if you've got very low levels of ghrelin, your disease will be worse. And that is reflected here, right? So the cluster 3, which is the severe cluster that I'm going to call for now, has the lowest level of ghrelin. Compared to cluster 2, the mild, has completely normal ghrelin. It doesn't even come up, right? So it's just great. Now, I mentioned earlier something about cell-to-cell transport. Now, this is a protein that's been flagged before in previous proteomic studies that is closely linked to cell-to-cell transport, syntaxin 7, and it's completely normal in the severe group, and it's the most reduced in the mildest group. Now, this kind of suggests, well, are there kind of certain cluster-specific or variance-specific vulnerabilities and what's the term I'm looking for? Kind of vulnerabilities and kind of protective elements or resilience factors, that's it, that people can express to kind of fight the disease. So one would hypothesize, for instance, that if you're in cluster 2, you've got milder disease, you've got higher resistance to the disease, and therefore you downregulate this protein to reduce the amount of cell-to-cell transport that's happening, and therefore reducing the disease progression. This is purely a hypothesis, but it kind of gives you an idea of how we're trying to understand and unpick these results. Right, and then the other ones that I'm kind of highlighting are kind of in the similar vein. So this osteoclastin, which is the BTLAP, is a protein that's been linked to Parkinson's disease, and it's the highest in the worst disease, and it's the lowest in the mild version of the disease. Right. So, as I kind of put here in the take-home message, there are some protein level differences between PD subgroups, and they highlight the potential biological differences within the PD kind of realm. Right. Now, as I've touched on before, every person's journey with PD is different. And every person reaches their PD milestones at a different time point, right? Some people need a walking aid earlier, some people start freezing earlier, some people never freeze, or very, very late in their disease course. So now it kind of comes back to the second point that I mentioned at the beginning of this talk, which is prognostic biomarkers. Are there proteins that can be measured early on in disease, right, when you enroll with us at visit 1, right, within 2 or 3 years of your diagnosis? And if we measure that protein, can that predict or give us an idea of your risk for a future PD milestone, right? So, for example, can we predict the risk of you developing dementia? Now, we know that dementia is a common complication in Parkinson's, and it's typically a late complication. This is from a Swedish registry where they looked at 1,500 people with Parkinson's just in the community. They followed them up over 20 years and then compared them to healthy controls, and which are age and sex matched. And you can clearly see that if you've got Parkinson's, your risk of developing dementia increases over time, right? So about 50% of people at the 20-year mark have developed dementia, right? And this is largely reflected on our cohort as well. So you can see here our follow-up is up to 12 years, and roughly about 65% of people are disease-free. So in other words, about 35% of people have developed dementia at this time point. So can we predict this? Can we use proteins to kind of predict your risk of developing dementia? And in short, yes. From our data set, we are able to identify one protein, which is called GFAP, or glial fibrillary acidic protein. And this is a protein that was quite a aha moment when it kind of came up in the analysis, because this is a protein that is very strongly linked to neurodegenerative disease and kind of brain injury as a whole. This is a protein that's elevated if you've had stroke, you've had traumatic brain injury. It's been linked to Alzheimer's disease. So it's a highly brain-specific protein, right? It's unlikely to be a noisy signal. And it's been once before described in the last 2 years in a fairly small Parkinson's study that also did proteomics, and they also identified this protein, but it was not longitudinal. They didn't look into the future, they just looked at people with dementia and without dementia in PD, and they found that this GFAP was high. So the fact that we were able to pick this out is really quite promising. And what this plot demonstrates is really just visualizing this risk. So green means you've got low GFAP at the time of we take the blood test, right? Which is early on in your disease, years from having any cognitive impairment. So that would be green. Yellow is kind of a medium level of GFAP, and red is a high level of GFAP. And you can still hear me, yes. And as you can see, the risk of developing. Have I lost you? Yeah, there we go. The risk of developing dementia becomes greater and greater and greater at a faster pace if you have high GFAP levels in the beginning, right? And another way to kind of sanity check this is to say, well, let's split up the people that go on to develop dementia later in life to the people that never develop dementia later in life, all right? At least as far as we know, right, for the period that we followed them up. And you can see at baseline, at the time of measurement, they already had high GFAP levels. So this, really, in summary, tells us that higher blood GFAP levels in early PD results in a doubling of the dementia risk. That's roughly about 2.5 increased risk. And really proposes GFAP as a very promising prognostic biomarker in Parkinson's. Now, what are our next steps? So we want to further untangle the proteomic differences between PD subgroups, right? There's clearly some hints, some interesting results that we can already kind of glean, but there's more work to be done. And you know, the cluster, the four clusters that I mentioned earlier, that kind of tries to stratify disease severity, is just one way of looking at heterogeneity in PD, and there are many different ways of looking at it. So there's many approaches still to take for us. We want to search for other proteins that are linked to PD milestones, not just dementia, right? So your risk of needing a walking aid, having falls, developing freezing. And we want to try and identify a proteomic signature in the RBD group that we haven't looked at as intensely yet, that might be able to predict phenoconversion. So in other words, your likelihood or your risk of developing PD in the next 5 or 10 years. So, in summary, we've identified shared alterations in immune activation and cell-to-cell transport in PD and RBD. We have proteomic variability between PD subgroups, hint at kind of biological differences in the drivers of disease. And at the level of a single protein, measured shortly after diagnosis, denotes your risk of future dementia. I want to thank you, you first of all the participants that made this project possible, and then also my team, and Professor Michelle Hewins specifically. And thank you very much. That's the end of my talk.